Life Science. Today we're going to start a new chapter of Part 2 Immunology, Chapter 7, the Immune Response, Document 1, Nonspecific Immune Response. The scheme you see in this slide is the same we have explained previously in the previous video. I'm going to start with this scheme since it summarizes the main steps of the immune response we're going to see in this chapter. So please pay attention. First of all, what are natural barriers? Natural barriers are parts of the body that may prevent the entry of the antigen. For example, the skin, if it's not damaged, it prevents the entry of the microbe into your body. Okay. Suppose that the natural barrier, like the skin, is damaged. What happens? Then there is infection. The non-self enters the body. Example, a virus or a bacterium. Okay. First of all, a first immune response is triggered within few hours after the infection. So this is really rapid. What is this immune response? It is the non-specific immune response. We consider it as the first internal line of defense. It's immediate because it's rapid, and the effector of this response is the macrophage. How? This macrophage is going to eliminate the intruder by a mechanism called phagocytosis. Then, if the macrophage fails to eliminate the non-self, here another immune response is triggered called specific immune response, and it's the second internal line of defense. This immune response is triggered within several days, so it's somehow slow, but actually it's more efficient than the non-specific immune response. The effector of this specific immune response are the lymphocytes B and T. Let us start first with document 1, non-specific immune response. So please open your books to page 138. Before we go deeply into the non-specific immune response, let's first see what are the natural barriers of the body. These natural barriers are the first line of body defense. They protect it, and they prevent the entry of invaders, and consequently, they prevent infection. Humans are vulnerable to invasion by pathogens in the air, food, water, soil, and also during physical contact with infected people. That's why natural barriers are very important, especially to protect us from infection. As you see in the figure, the most comprehensive barrier is the waterproof layer of the skin that covers the body's surface. The human skin keeps most pathogens out as long as it remains intact. Other barriers are the mucus, the tears, saliva, sweat, acids in the stomach, the commensal bacteria of the gut and the genital tract, the antibacterial proteins in the semen and in vaginal secretion. The commensal bacteria of the gut, they are friendly bacteria that live in the digestive system, and they compete with the harmful bacteria, preventing them from causing infection. The inner surface of the respiratory system, of the nose, throat, airways, and lungs, also the stomach, the intestines, the bladder, the reproductive tract, all these parts of the body are lined from inside with membranes that secrete jelly-like mucus, and this mucus actually It presents a barrier against pathogens entering our tissues via water or food or air. And that's why you see in this figure the mucus and the intact mucous membranes of the respiratory system. Also, you see the mucus and the intact mucous membranes of the digestive system. You see also the cilia in the respiratory system. These are structures that actually found on the surface of cells at the level of the respiratory system. These cilia, they trap foreign material and they push them upward, preventing the infection of the lungs. These natural barriers are divided into physical or mechanical barriers and chemical barriers. The physical barriers include the skin, the vibrating cilia of mucosa cells, the mucosa of the respiratory, digestive, and reproductive system. While the chemical barriers consist of the sweat for the acid it contains, and this acid can destroy bacteria. The gastric secretions, also the gastric juice, it's acidic. The tears, they contain enzymes that can destroy some bacteria. The nasal mucus also. The saliva for the enzymes it contains. And the semen and the vaginal secretions for the antimicrobial proteins they contain. Okay. As we said earlier, that if the natural barrier is damaged, then this will stimulate a first immune response to be triggered. It is the non-specific immune response. It appears very quickly within few hours after contact with the antigen, but it has low efficiency. Who is the effector of this response? It's the macrophage. And why is this response considered non-specific? We're going to see that it is independent of the identity of the intruder. How is this response manifested? By a process that we call inflammation. What is inflammation? So, a manifestation of the non-specific immune response is inflammation. So when the natural barrier is damaged, for example the skin, the non-self enters the damaged tissue and triggers a non-specific immune response expressed in the form of inflammatory reaction. This reaction, called inflammation, is characterized by the following symptoms or signs: redness and heat, edema or swelling, pain. These same signs appear in the case of many infections, such as dental abscess, pharyngitis, which is the throat infection, etc. The physiological events leading to these signs are broadly referred to as inflammation. What are the physiological events that lead to the appearance of these signs: redness, heat, edema, and pain? The figure below summarizes the physiological events of inflammation. Please look at figure 1. When bacteria enter a tissue, the infected cells that are present in the tissue and some white blood cells release a variety of chemical messenger called cytokines. You see them in blue, the blue stars. Okay, these are small soluble molecules that act on different target cells, and they have local and systemic effects. What are the local effects of cytokines? Let's see. If we compare figure 2 to figure 1, we can maybe draw out these effects caused by cytokines. What do you see inside the blood capillary in figure 1? You see you have the monocyte with horseshoe-shaped nucleus. You have the lymphocyte with round nucleus. Of course, all these, they bath in the plasma. Okay, by comparing figure 1 to figure 2, can you see any changes? Yes, there is a local enlargement of the blood vessel's diameter. And this leads to an increase in the blood flow to the inflammation site. So the blood vessel's diameter increases and more blood flows to the inflammation site. What else can you see here? Yes, there is an increase in the vascular permeability, and there is leakage of plasma from the blood vessel into the surrounding inflamed tissue. So you see fluids and proteins are leaving the blood vessel and moving toward the inflamed tissue. Actually, you see also that the white blood cell, the monocyte, is squeezing itself between the cells of the capillary wall. It is trying to leave the vessel, right? Actually, some cytokines are chemotactic. What does that mean? These cytokines, they attract leukocytes, and mainly the phagocytes like the monocyte, to the site of infection. So the monocyte crosses the permeable blood vessel and migrates into the inflamed tissue. The process of migration of this monocyte outside the blood capillary, this is called diapedesis. Okay, all these changes that you see in figure 2 are caused by cytokines. Increase in blood vessel diameter, leakage of plasma outside the blood vessel, and also the leaving also of the monocyte to the inflamed tissue. In figure 3, you see how the monocyte now is outside the capillary and it has transformed into a bigger cell called macrophage. Also, the granulocyte with yellow nucleus is trying to do the same, and it was also considered as a phagocyte. These phagocytes now are preparing themselves to digest the non-self. Okay, we said that after infection, there is inflammation, and the main signs of inflammation are redness and heat, pain, and edema. Okay, how can we explain these signs of inflammation? Let's relate the physiological events that occur during inflammation and the signs of inflammation. One of the local effects of cytokines was the dilation of the blood vessels and the increase in blood flow. And actually, that's what causes redness and heat. What about swelling? The increase in the permeability of the blood vessels and the leakage of plasma outside the blood vessel actually causes swelling and the accumulation of the plasma outside the vessel under the skin. What about pain? The compression of the tissue caused by the swelling, as well as the excitation of the nerve endings by the cytokines, cause pain. So, the local effects of cytokines that are dilation of blood vessels, the increase in the permeability of the blood vessels, and the compression of the tissue caused by swelling, these local effects of cytokines are the main cause of the signs of inflammation that are redness and heat, swelling, and pain. Also, cytokines have systemic effects, and the main systemic effect is the increase in body temperature, or the fever that you know is the main effect that happens during infection. Okay, to finish this document, let's see how the macrophage or the granulocyte undergo phagocytosis to stop the infection. So what is phagocytosis? Phagocytosis is the process by which phagocytes, like granulocytes and macrophage, digest and eliminate the non-self and thus stop the infection. In this case, the nonspecific immune response succeeds in eliminating the intruder and protecting the body, of course, if phagocytosis happens. So, after infection, the monocyte leaves the blood capillary and transforms into a bigger cell called macrophage. Similarly, the granulocyte is able to leave the capillary in order to eliminate the intruder. Granulocytes and macrophage are called phagocytes. Phagocytosis consists of 3 steps. Look at the figure, please. Step 1, adhesion. And during this step, the white blood cell, the macrophage, of course, or the granulocyte, approaches the microbe, adheres to it, and engulfs it. In the second step, absorption, the microbe is absorbed by a vesicle of endocytosis. In the third step, the ingestion, the lysosomes fuse with the vacuole of endocytosis. As you see, the microbe is inside and the lysosomes surround the microbe, and we have formation of the phagocytosis vesicle, or that we call also phagosome. Here, this is the ingestion of the microbe, and digestion might follow. What happens? 3 possible outcomes. In the first case, the enzymes degrade the microbe and the wastes are eliminated. So, the infection is stopped. The enzymes, of course, they are found inside the lysosomes that surround the microbe. Case 2, in case of dysfunctional enzyme, the enzymes are non-functional, inactive, or they are very few, or resistant bacterium. Also, the microbe could be resistant, strong. So, the microbe is not degraded. It remains, in this case, intact, dormant inside the cell. And this is what we call a stationary state. In case 3, the microbe can multiply later on. And you see that the nucleus of the macrophage or of the granulocyte is destroyed. It disintegrates and there is formation of the pus, which is a sign that the macrophage, the white blood cell, is destroyed. And of course here, infection is continued and the nonspecific immune response has failed. What's going to happen? Of course, there will be a need for a second immune response to interfere, which is the specific immune response. And that's what we're going to see in the next video. As a conclusion, the natural barriers are the first line of body defense, like skin, gastric juice. The nonspecific immune response is triggered only few hours after infection. The 2 mechanisms of the nonspecific immune response are inflammation and phagocytosis. Thank you, dear students. Stay safe and see you soon.